Have you ever wondered about how DNA replicates itself?
Wonder no longer.
Today, we'll walk you through the structure of DNA and the various steps it takes when replicating itself. Well, the first question you might be asking is, what is DNA made of? To better understand DNA, let's think of it as a twisted ladder. Each rung of the ladder is made up of two bonded nitrogenous bases.
These are either adenosine and thymine, which connect using a double bond, or guanine and cytosine, which connect using a triple bond.
It is crucial to note that the nitrogenous bases always pair up in this fashion. The sides of the ladder are commonly referred to as the backbone of DNA and are made up of alternating deoxyribose and phosphate molecules. The sides of the ladder are said to be anti-parallel, meaning that they run in opposite directions. This means that when the ladder side has a phosphate on the end, it is known as the 5' end, and when it ends in a deoxyribose, then it is known as the 3' end.
The second question you may be asking yourself is, how does DNA replicate? The first thing that needs to happen for DNA to replicate is for the two strands of DNA to separate. Topoisomerase unwinds the double helix, and helicase exposes the individual nitrogenous bases on each of the strands of DNA by breaking up the hydrogen bonds that bond the nitrogenous pairs together.
The result is two halves of DNA that can act as templates in the next steps. Next, RNA primase places a few RNA nucleotides and binds them to the template strand.
The job of the RNA nucleotides is to act as the foundation for the building of the rest of each new strand of DNA. The new DNA nucleotides float around in the nucleus. Some extra phosphate ions are attached to them. These extra ions break off and provide energy to the reaction. The DNA polymerase II moves the nucleotides and positions them along the separated strands using base pairing. It also acts as an exonuclease by checking the placement of the nucleotides and removing those that were placed incorrectly. DNA polymerase I replaces the RNA primer pieces left by RNA primase with DNA nucleotides. Also, if the bonds in the backbone break for any reason, ligase goes in and corrects them.
So, next question is, what is up with the legging strand? Helicase unwinds the helix in the direction of 5' to 3'. The DNA polymerase II can only build DNA in this direction as well. Resultantly, one strand of DNA is in the correct direction and one is backwards. The one that runs in the correct direction, which is 5' to 3', is the leading strand. And the one that runs from the 3' to 5' end is the lagging strand. On the lagging strand, the DNA polymerase II builds in small chunks, going backwards each time, to start over from the 5'. These chunks are called Okazaki fragments and need to be filled in with RNA primer. The RNA primer pieces are later replaced by DNA polymerase I.
And that's how DNA replicates.
Thanks for watching.